Now try it.
Oh, okay, okay. I do have some disclaimers because I do clinical trials for several drug companies, and these are the ones that I'm currently involved in. But are you having trouble hearing? Okay. Let's see. I'll stand closer to the mic. Is that a little better? Okay. This is the group at the University of Texas, Houston, our Scleroderma Center, all of the physicians, laboratory people, nurses who are involved. And it's because of these people that I'm actually able to do this research. But let's talk about an overview of research in general. There's basic science research, which is really laboratory research. This has to do with mice and cells in the lab. And then there is translational research that takes laboratory research, basic science findings, and takes them into a situation in which they can be hopefully beneficial to patients in the clinic. It translates from the lab to the clinic. And then there's clinical research. Once we found a drug or a procedure that we think is going to be helpful to patients, this is when we do the clinical trials to see if in fact what we think, what happens in the test tube and what happens in mice also will happen in human beings. Well, if you look over the years, this is, let's see if I've got, well, you can see that. I don't think people virtually can see, but anyway, this starts in 1940 and ends in 2023. This is the number of publications that lists scleroderma as one of the key words. And this is what is published in an online scientific compendium put out by the National Institutes of Health called PubMed. And I think many of you may have gone online because it's open to anyone. You don't have to be a scientist or a researcher. And key in the word scleroderma, and in 1940, there would be one article coming out. And then, whoops. And then, let me go back for just a second. So if you look at, this is 2023, of course, we're only halfway through, but you can see that there has been a steady increase in the number of publications. Each of these represent some laboratory or clinical finding that is important to scleroderma. So what has basic science taught us about scleroderma in those years from 1940 to 2023? Well, if you look at immunology, vascular changes, fibrosis, and genetics, we've learned a lot, but of course, what we don't know is what's the trigger, what's the cause of scleroderma, and how can we cure this? But nonetheless, we've learned a great deal in those many years, those many decades. In terms of immunology, there are multiple types of immune cells that interact in complex ways. And that makes it more difficult to treat because there is no one type of immune cells. There's not one type of B cell, one type of T lymphocyte that is so important to scleroderma that if you block that and block that alone, you can reverse the changes, stop progression, cure the disease. All of them work together, and that's all part of the human immune system. And in terms of vascular changes, there's small blood vessels that are affected early, and this is what causes Raynaud's phenomenon, which I'm sure most of you are way too familiar with. The small blood vessels, the end blood vessels in the fingers and frequently in the toes as well, become narrowed, fibrotic, damaged, and they're responsible for all, but not for some, but not all of the damage that we see in scleroderma. And the other part, the immune part, and then there's the vascular changes, and then there's fibrosis or scar tissue that, whoops, hit the wrong button, that builds up in multiple organs, the lungs, the GI tract, other organs, that works triggered with the vascular changes and immune cell stimulation, and sometimes will continue to progress, and sometimes on its own will progress to a certain point and then stop. Not all scleroderma has to get worse. And then there's the genetic component to scleroderma. And now there are over 30 genes that we have identified that contribute to susceptibility. So when I started genetic studies some 20 plus years ago, this is when DNA was going to answer all of our medical questions. And I thought, well, maybe there's one gene or a couple of genes, and we will identify those, and we'll find the main trigger for scleroderma. Well, we found several genes, and then the more we looked, the more we found, and now there are over 30. And these genes work together. Not one by itself is responsible, or two or three by themselves. It's a combination of genes, and it doesn't cause scleroderma. It just increases the susceptibility. It increases the likelihood that any individual will get scleroderma, but it has to be in conjunction with immune changes and vascular changes and these other things that go along together. So moving on to clinical trials that I know people are very interested in, and for any individual patient with scleroderma or their family members, this is really where the rubber hits the road. And so if we take everything we've learned in the lab, in mice, and we see, will these findings translate into help for scleroderma patients? Well, there's a phase 1 trial when a new drug is identified as potentially helpful. And the question in a phase 1 trial is it safe in humans? And how should the treatment be given? Is it a pill, which would be, of course, the easiest thing for people? Does it have to be given as an injection? Does it have to be given as an IV infusion every month or so? And what's the right dose for a person? And a phase 2 trial looks at the effectiveness to some extent, but mostly it is a safety trial looking for side effects. And then in a phase 3 trial, and this is now the bigger trial, determine whether the new treatment is a better alternative than the current standard of care treatment. Because even though a medication may be helpful, if it's no more helpful than mycophenolate, Cellcept, or other medications that are out there approved and used, then it's not really going to be all that helpful. And then there's a phase 4 that I'm not going to be talking much about. That's post-marketing. In a rare disease like scleroderma, but it's not rare in this room, it's not rare in this conference, it's not rare in my clinic, but in a rare disease overall like scleroderma, just because you try it in 200 or 300 patients in a phase 3 clinical trial, maybe there is a side effect that only happens 1 in 1,000 patients. You're not going to find that. You're not going to identify that in a small trial. So sometimes the FDA, which is the approval agency in the US, I don't know, I guess it's decided that it's a little too warm in here, so they're turning on the fans. Oh, it's raining. Oh. Huh. Well, okay, moving back. In post-marketing, the FDA will require that the companies who are making the medication follow up on a longer term as more and more patients get prescribed the drug. Well, now this is an old slide as of January 2015. The pharmaceutical research and development process. So this is the preclinical all the way to your left. This is when the mouse studies and the cell culture studies, they can identify certain medications, hundreds, sometimes thousands, and then it goes through the phase 1. Is it safe? What dose will we use? The phase 2, looking at several more patients. The phase 3, which is really the larger study that will be presented to the FDA to see if they're going to approve it. So out of all of those, and usually millions of dollars spent by the pharmaceutical company, we may or may not have a drug that is helpful. So let me talk about some of the general design of almost all clinical trials. Usually they are controlled, which is to say, some of the participants in the trial get the new medication and some get a dummy or a placebo medication. In most current scleroderma trials, participants are permitted to take their usual or background medication. So we add the new medicine to it, and the advantage of that is that people don't have to stop what they're taking, which may be helpful, but maybe not as helpful as we'd like. They can stay on their background medication, and we add the new medicine to it. And it's randomized. So whether or not you get the new medicine is determined by chance. And usually these days, we type some things into the computer, and the computer says, well, this is patient 001, and they're going to get the bottle labeled, 1234 dash whatever. And from then on, for the duration of the trial, that's their trial number, and that is the medication that they have been assigned to. And you don't know, and I don't know, as the principal investigator of this study, whether that's the active medicine or the placebo. So this is studies I have been involved with from 1995 to about 2016. You can see the list there, the D-penicillamine trial, a photopheresis, interferon, all the way down to cyclophosphamide study, the lung study 1 and 2. Well, what happened in all of these trials beginning in 1995? Oops, I think we are missing. Well, the penicillamine trial was a negative trial, which is to say that the people on the true medication and those who were assigned to the essential placebo arm, there was no difference. At the end of the 1 year of study, some people got better, some people got worse, some people stayed the same, and there was no difference between what we thought was an active medicine and what the placebo was. Then there was a photopheresis that was -2. An interferon study was negative. And as you go beyond that, you can see negative trial, negative trial, negative trial. And we kept thinking, we kept doing trials because we thought some has to work. Some medication somewhere, keep on doing it. We didn't have much to offer our patients back in 1995 to 2000. And finally, the Scleroderma Lung Study 1 was a positive trial, and that led to Lung Study 2, also a positive trial. We're going to talk a little bit about that. But before we do, I'm going to go back and make sure you know who pays for the clinical trials. This is very important. It can be a government agency like the National Institutes of Health. And for those of you who were here, when the conference started yesterday evening, and Dr. Lindsay Criswell, who is now the director of the NIH branch that funds clinical trials in scleroderma called NIAMS, she talked about the amount of money that they have contributed, and we're all very grateful. Then there is the DoD, the Department of Defense, and many of you may not be aware that the Department of Defense also has an arm that funds medical research. And And the way this started, I always thought this was interesting, but the way this started was that the Army wanted to get medication for tropical diseases, malaria and other diseases that the soldiers and the military would be exposed to, so it was helpful to them. And also to do studies of battlefield wounds, how best to care for them. And then Congress, through the advocacy of others, added different diseases, one of them being scleroderma. So they now fund some scleroderma research. And then there are foundations and other organizations, obviously the National Scleroderma Foundation, Scleroderma Research, there's a Scleroderma Clinical Trials Consortium that I'm a member of that also is very interested and very active in doing research. And then there are the pharmaceutical companies, which are actually the mainstay of funding for clinical research. So these are the negative trials. So positive trials from 2016 to 2023. There's the Scott trial, which some of you may be aware. This is the high-dose chemotherapy followed by stem cell transplant. And it is effective, but it is also, as you can imagine from the high-dose chemotherapy part of it, is quite difficult, quite toxic to patients. There was nintedanib, brand name Ofev, that at least one person in the audience was involved in. For scleroderma lung disease, it is now FDA approved, and this is industry sponsored. There was tocilizumab, another medication in scleroderma-related lung disease, also FDA approved. There's another one, pirfenidone. It's also for lung disease, but it is not FDA approved for scleroderma-related lung disease, but for another similar lung disease. And then there are multiple drugs that have been tested and approved for pulmonary hypertension, which are also helpful for the Raynaud's, but it's not approved for that indication. And then mycophenolate, which is also known as Cellcept. Now this is not FDA approved, but it is Medicare reimbursable. Yes, and I can see some of you wondering what the difference is. In order for something to be FDA approved, it has to go through these clinical trials and an application has to be made to the FDA, and it is a very expensive process. In the meantime, mycophenolate went off patent. It became generic, and there was no longer industry interest in spending millions of dollars to get it officially approved. So just because your physician offers something to you that's not FDA approved does not necessarily mean it is wrong or inappropriate, because there's a difference between being standard of care, which mycophenolate Cellcept now is, and being FDA approved. So, recent positive clinical trials. Let's talk a little about the scleroderma lung study 2, SLS2. It was sponsored by the National Institutes of Health, the NIH. Before we get to that, and I'm not sure, I think many of people in this audience know what interstitial lung disease is in scleroderma, but just to make sure, we'll talk about it. It's scar tissue buildup in the lungs, and it usually starts in the bottom of the lungs and the bases, and then gradually works up the lung. Now, not all scleroderma patients who have the beginning or a little bit of scleroderma lung disease, not all patients will progress. And this is one of the reasons we still need to do placebo-controlled trials, because some of the people in your trial are going to get better or be stable even on placebo or on background medication. So you really need that extra arm, that controlled arm, in order to determine if something is effective or not. And how do we screen for lung disease? We screen by pulmonary function tests. And just because someone has a normal pulmonary function test today doesn't mean that 1 year from now it's still going to be normal, or 2 years from now. So it's a screening test. We need to do it. I usually do it on an annual basis. Some people will do it every 2 years, but nonetheless, it really needs to be done regularly to make sure that this individual is not developing scleroderma lung disease. And if the pulmonary function tests are not normal, do we and they have scleroderma, do we automatically say they have ILD? No, because pulmonary function tests normals vary a great deal, from 80% predicted to 120% predicted. So just because somebody is an 80% or even lower doesn't mean that they necessarily have lung disease, and you need a CAT scan of the lungs in order to really identify if lung disease is present. So this is a CAT scan, this is a chest x-ray of somebody with lung disease, and you can see that there is a lot of white spots through it. It's sort of hazy. Well, those white areas should not be there. The normal part of the lung, and in this x-ray, the only normal part of the lung is at the top where it is quite dark because air is radiolucent. It looks clear on a regular CAT scan, a regular chest x-ray or CAT scan. So CAT scans would take a slice of the lung. So rather than somebody standing for a chest x-ray, they're lying down, and the CAT scanner will go and do horizontal slices through the lungs. And the one to your left is a normal CAT scan with a lot of dark spaces, and the few white spaces that you see are from blood vessels in the lungs, and that's normal. And then if you look at the other CAT scan, you can see that there's a lot of white areas, and those white areas represent scar tissue that shouldn't be there. So SLS2 was a randomized, double-blind, prospective, controlled trial, 1 year of daily oral cyclophosphamide, which is a chemotherapy drug, versus 2 years of daily oral mycophenolate, Cellcept, and the primary outcome measure was the pulmonary function test at the end of the study. Now, this has become fairly routine in clinical trials with an outcome of scleroderma-related lung disease. And as a result, both treatment arms resulted in improvement or stabilization of lung function as determined by the pulmonary function test, CAT scans, a shortness of breath questionnaire, and skin scores. But the advantage was that mycophenolate had fewer side effects than daily chemotherapy, which I think all of you understand. Yes, you would expect it to have fewer side effects. And the basis of this, mycophenolate was considered a standard of care. And now most patients with scleroderma lung disease, early lung disease, will be placed on mycophenolate. So what other medications do we have for scleroderma lung disease? We have tocilizumab, which is brand name Actemra, FDA approved. There's also what I mentioned, nintedanib, also a pill, FDA approved. Oh, the tocilizumab is an injection that you get weekly or every 2 weeks. And then rituximab, which is an IV infusion that's given every 6 months. There are many reports now that suggest it's effective in scleroderma. It is approved in some countries, including Japan. It is not approved yet in the US, and may or may not be. But there are now multiple other clinical trials that are being looked at for scleroderma skin disease, scleroderma lung disease, because these are two organs that we can fairly readily measure. and you can't test a drug for an organ system that you cannot measure. So the next part I'd like to talk about is a clinical trial right for you or right for the scleroderma patient that you know? So there are several issues, the first one of which, are you willing? And the second, are you eligible? Just because somebody is willing to participate in a clinical trial doesn't mean that they are eligible, because there are a great deal of inclusion and exclusion criteria that you have to meet in order to be considered eligible. So all trials have different eligibility criteria. So you may be eligible for a clinical trial of one medication, but not of another. And most trials require early disease, and by early, by definition, we mean within the first 3 to 5 years of disease onset. Now, this is not disease diagnosis, because many of you still experience a delay of 1, 2, 3, or more years from the first time you have Raynaud's and puffy fingers, or trouble breathing, or increased pigment in the skin, or any of the dozens of symptoms scleroderma can cause. From the onset of the disease until diagnosis can be quite lengthy. And then your doctor says, oh, you may have scleroderma. I'm going to send you to a scleroderma center. So you call, and the appointment is 6, 7, 8 months later. So now you're 3 years, 4 years, 5 years from the onset of disease, and you may not be eligible for the trial. We, at UT, my wonderful research nurse reviews medical records of everyone who's been referred, even if their appointment is months away, to see if she can identify people who are likely to be clinical trial participants, and we try to move them up. And most trials require diffuse skin involvement. Now, you may have limited skin involvement, which is to say thick skin confined to areas below the elbows or below the knees, does not involve the chest or the abdomen or the thighs. And you have real scleroderma, and you may still have lung disease or other complications. But if you don't have diffuse disease, for a trial that is looking at diffuse skin involvement, you're not going to be eligible. And many trials, not all, require lung involvement. If lung function is the primary outcome, as the focus of the trial. And then there are laboratory requirements. You can have a little bit of anemia, but not much. Maybe you have a little decline in kidney function, but there are limits set to that, liver function, etc. So even though you may be eligible in 9 out of 10 criteria, if you're not eligible, if you don't meet all of the 10, then you're not considered eligible. So I'd like to ask the audience who has participated in a clinical trial. Okay. I'm going to start here. Which trial, if you don't mind saying? Yes. Yeah. So this is not a clinical treatment trial, but it is what we call translational research, where the skin biopsies are taken to the lab and multiple kinds of tests are done, and we extract some of the cells, extract some of the DNA, and try to identify changes that are specific for you and generalizable to other scleroderma patients, to try to say, this for the purpose of individualized treatment that for this patient, it looks like this metabolic chemical pathway is activated. So maybe they would respond to a medication that addresses and targets that pathway, whereas somebody else with a different pathway is not going to respond. So we should use something else. So yes. Yes, yeah. And that was a clinical treatment trial. Yeah, yeah. So I know it's, yeah, we're going to have to speak a little louder. Yeah, yeah. And let's see, there was someone else who was part of the trial. I'll try to repeat for the people online what your experience was. Oh, the Celgens trial. That was the extraction of, yes, yeah, immune cells. Yeah, that was a trial I had not listed. But yes, there's, yeah, yes, uh-huh. Yes. Yeah, yeah, and most of the trials now, even the pill trials, are also requiring skin biopsies before and then at the end of the trial. Yes, and so how much, pardon me? Yes, yeah, so what, let's see, can we, I'm not sure this, I can, oh, yeah. Okay, well, maybe we can, but I won't be seen anymore, so we're gonna have to have a volunteer come by, and, okay, we have a volunteer who will, okay, that's fine, thank you, because I would like to get this. So the question to you is, yeah, what was your experience with the trial? Yeah. Yes. Right. Louder. Yeah, or closer. Good. Good.
So every week the dosage removes.
Yeah.
And then went to see how I tolerated it. So I went to, yes, I think it went up to 3,000.
Yeah, as a total. Yeah.
But it helped. My cough went away.
Yeah.
And it really helped.
Okay. Yeah, good. Why did you join the trial? Why did you go? Why did you participate to begin with?
Oh, I mean, I was coughing and swollen. Instead, the option of a lung transplant was
Yeah.
there. So this was, I wanted to be in it.
Okay, and if you could bring the microphone over here. Yeah. Okay, so your experience in the trial, and you have to hold it close.
My experience has been great. I really haven't heard it's loud.
It'd be better for you because if you're passing the microphone.
My name is Rose Gonzalez Pettis, and I'm from San Antonio, Texas. I'm really thankful to Dr. Ray for letting me speak today because I just got into the clinical trial after patiently waiting and returning multiple times over the course of the year. And I was fortunate to get into it because a girl who I babysat and played with 2 doors down in my neighborhood in Mathis, Texas, small town, only 4,998 people there. We both got it, and it was a shock and rare because nobody had even heard of it before. And so she was fortunate to meet Dr. Mays and get into a trial. And so I was invited to come and to see Dr. Mays, and my experience has been great. It's been, at the beginning, lots of tests, and actually amazing to go through the Health Science Center and Herman Hospital and meet doctors that are doing research that you just couldn't even imagine all over the world. And honestly, I was being very selfish because I wanted to get cured, because at one point I couldn't sit and stand, and it seemed like every time I went to Mathis, I'm from San Antonio, but when I go to Mathis is when the symptoms would appear. It's also when I'm at a high stress level because that's where I do my work. And fortunately, Dr. Mays told me to get on mycophenolate, and so I did, and that helped. And now I've been accepted to a clinical trial study, and I'm taking 2 pills in the morning and 2 pills at night. I call them my silver bullets, and I'm taking the real thing. I know I am. And then I got asked to be in a second trial with Dr. Sam Theodore, similar to what my friend Peter Mann described. And I am just pleased because once I said I want to do this to help others, that's when good things happened. And I'm a very strong woman of faith, and some very good things have been happening to me. And I'm very grateful to be a part of this and to be a part of 2 studies. I thought I couldn't do that, but one is medicine and one is not, so that's why it is allowed.
Yeah.
So it's been a great experience, and I'm even going to get to do it at home. The next 2 visits, a nurse will come to my house if I want to travel to Houston from San Antonio, and she'll do some of the tests in my home, which is absolutely amazing. And you get paid for being a part of it. You get paid for your mileage and your meals. So it's really great, and it's really easy. And Dr. Mays cares and listens and is so patient. And before you even get in your car to leave, all the notes are already on the portal for you to read with your information. It's amazing. She's magic, and she's marvelous and doing wonderful things. I'm very proud of her for the 25 years plus of work that she's doing, because of her, great things are going to happen for us and people in the future.
Well, thank you. And I didn't pay her for that. Yeah, and one thing I wanted to say as well, and I have Ms. Perez's permission to say this, that she came for one clinical trial and was not eligible. But when you look through it, she was actually eligible for a different clinical trial. So we were able to offer her something for all of the time and effort and energy that she spent, and the generosity in being willing to participate. Yes, you had a...
Yes, I've been coming to you for a number of years, and you've given me compliments. But I've been talking about something I might need some, maybe some trials or something, because of the bronchiolitis. Have you heard of that?
Yeah, that's an
Increased mucus production.
well, it's affecting not just the sort of the air sacs in the lungs, but the small air passageways that go to the air sacs that get inflamed and affected. Yeah. It tends to be a side effect. But I think this is a great example of someone who has been stable for a period of time and doing well, and then the disease can flare up again and worsen. And so it's a matter of always being vigilant, of doing the pulmonary function test on a regular basis. I don't do CAT scans over and over again because it's a little bit of radiation, and if the pulmonary function tests are stable and people are doing well, I really don't see a reason to take the risk of even a small amount of radiation. And these days, CAT scans use very small amounts of radiation, so they are very safe, but you still don't want to do them over and over again.
Do you need the blood test?
Yeah.
And the mucus clinic, or the doctor said that it comes from hardly even of us better.
Right, right. Okay, well, and others, because not just, yes, we have someone in the back.
So about 6 years ago, I was in a study for
Oh, you have to bring the mic a little closer? Yeah. Yes, it's better.
So I was in a study for drug, and it did not want to have a positive ending, but at first I thought it was doing some good, and that was a lot of things. Walks, and very hard casts, blood tests, function tests, all those things. And the reason I didn't was I did it for hypertension. But I am a researcher, a scientific researcher myself, but not in the medical field. But I feel that we really need symptoms to do this. And I think the more you do it, but seek it, even if you don't absolutely need the drug, but to help with this research, the better it's going to be. Yes.
Well, thank you. Thank you. And I appreciate to say, this may not help me. I end up on placebo, but I'm going to do it to help people who come after me. Yeah.
Actually 16, and so he's not in and stuff. But with your have you seen he is on Actimra, he is on he is on Silsa. Have you seen patients that are like on trials, seen improvements with MPH? Because I question whether we need all four or we don't need all four. Have you seen them have improvements? I have seen, since he has been on that Actimra, I have seen amazing things with his lungs. We've been having a lot of lung issues, and we are not having as many of those right now. So I have seen, since he's been on there, a difference, and it has slowed down his scleroderma. So do you think that these patients need these multiple drugs together, or is it something that, I don't know, in your trials, have you seen a lot of the patients that you're in your trials have these multiple medications taken together?
Yeah, the multiple medications tend not to be in clinical trials because you're permitted to be usually on one, but not on multiple. But I think what you are describing is an excellent point, that some patients require a lot of medications. But I've found that over time, they frequently, the disease activity will frequently level out, and that very slowly you can remove one or lower the dose of one, see how low you can get it, and maybe you can get people off it, and then gradually change things like that. Because what you don't want to do as a treating physician is to have somebody be fairly well controlled, even though it takes a lot of medication, and then you slowly lower and perhaps stop one, and then the disease flares again, and then it may be more difficult to get it under control again. And so, a gradual approach is it, but you're also right that for all of these trials, I'm an adult rheumatologist, not a pediatric rheumatologist, but for all of our trials, people have to be at least 18 in order to participate. So that means that people, and teenagers who are immunologically, physiologically, pretty much adults, but nonetheless, because of many legal and other issues, would not be eligible for a trial. But depending, the way that he can be on Actemra is because of clinical trials. Because people said, okay, I'm going to take the risk of being on a placebo injection. So one thing is a placebo pill, but it's a total other thing to say, oh, weekly injection, and this may not be the real medicine. That, I think that takes a very courageous person to say, okay, sign me up. And it's only because people were willing to do that, that medication is now available to your son and available to lots of other patients.
We have a couple questions from our virtual audience. If
good.
I can ask those. The first question is, how close, if at all, are we in the U.S. to getting Rituxan approved for SSC?
Oh, I don't have a solid answer to that because I'm not familiar with where it is in the approval process. And so I really can't address that. There are multiple studies that have indicated for certain subsets of patients it can be helpful. And it's just a very difficult question to address. You know, when I said that mycophenolate, which is Cellcept and Myfortic, the way that is paid for by Medicare, although it is not FDA approved, was through the efforts of a pharmacist called Jessica, what is Jessica's last name? Farrell, who gathered up all of the tests, all of the studies that have been done, applied to Medicare funding agencies, went back and forth, and it took 2 or 3 years, but it was through her efforts that mycophenolate is now Medicare reimbursable. And once Medicare agrees to pay for something, usually the private insurers will follow suit. So really, rituximab may not be FDA approved in the future, but it could be approved and for reimbursement by the insurers because of evidence that it is effective.
Thank you. There is a follow-up question of what concerns, if any, do you have regarding the danger of any immunosuppressive therapies for those with SSC and ILD, given the ongoing danger posed from not only COVID, but also RSV and other respiratory issues?
Do I worry about it? Absolutely. It is also helpful for people to be vaccinated against, well, COVID, obviously, and now other vaccines are coming out, but it is clearly a worry. These are immunosuppressants. The ability to fight infections, viral infections, bacterial infections, funny infections, run-of-the-mill infections, that ability is diminished. And I know I've seen people in this conference still wearing masks, and I think that is a good idea if you're going to be out in public exposed to a large number of people, then it's helpful to do all you can to try to protect yourself. But it's always, all of life is a risk-benefit ratio. So yes, there are risks to this medications, but what are the risks of not taking it? If you are getting worse and your lung function is declining, is it better to take the risk of the medication than not? And that's a very personal decision. Participating in a clinical trial is a very personal decision. Some people just get too anxious to think about being on a placebo for a year, make that commitment, and for them, a clinical trial isn't worthwhile. It's not for them, but for other people who are willing to take that risk, and frequently in a clinical trial, after the 1 year after the phase 2 or 3 part of the trial, if it's shown to be safe and relatively effective, there may be an open label extension. So after a year or 2 years' time, you can get the medication for free as a continuation of the trial. Okay. I think we have some time, yes.
You mentioned about you have to be 5 years or under for the trials. Are there any trials that you do for beyond 5 years?
Currently, I think there's one trial for 6 years, but I think your question really is, for people with more long-standing disease, is there a clinical trial? Probably not. We did a trial of a medication for Raynaud's, and for that particular indication, didn't make any difference how long you had scleroderma, as long as you had severe Raynaud's and so many attacks a day or so many attacks a week. So for particular complications, there may be a trial, but not for lung disease, skin involvement, and currently we don't have any treatment trials available for GI disease, which is something all treating physicians with scleroderma patients find very difficult, and patients experience very difficult problems. But if a medication is shown to be safe and effective, then it is usually not limited to people in the first 5 years of disease. It's wide open. People with longer standing disease can take it, and likely it will be effective for them as well. Yes?
Seems like this answered my question. I was going to ask about GI studies, the future of them, and where the status is of them. Because for me, that is my biggest struggle. It's not the jail all the way down. And the medications help. It's a huge quality of life problem. So you're working and having a social life and all of that. So I would love to participate in research. I've done a number of different registries, translational research studies. I'm happy to continue to do that, but I'd really like to see more on the GI side. So if you can talk about that a little bit more.
There are single center trials, usually open label, for GI involvement. One of the problems in doing a GI study is to measure the outcome. You need a number to measure. Like pulmonary function tests, that's easy. Forced vital capacity, 70% improves to 80%, worsens to 60%. That's easy. And skin scores for diffuse skin. Yes, your skin score started out at 30 and now it's 20, and that's a win. But how do you measure the motility in the GI tract? There are questionnaires that are very good and shown to be reliable, but it's not quantitative. And as long as it's not quantitative, it's unlikely to be FDA approved. So that's the issue. Now people are looking toward quantifiable measures in scleroderma GI disease. And if we can get that established, then we can do studies. And I think there was a comment. We've got about
4 minutes.
4 minutes.
Questions.
If you were designing a trial, I'm willing to show that the treatment work was scleroderma potentially versus is there a mechanism that you would use that you feel best, or
Yeah, well, there's an outcome measure called CRIS, the Combined Response Index in Systemic Sclerosis, that gives you certain points for lung involvement, better, worse, GI involvement, better, worse, Raynaud's, digital ulcers, all of that. And that's the closest thing we have. But right now, the FDA is looking at it favorably, but still, CRIS alone would not get a drug approved. Right. And part of the difficulty, of course, as the people here demonstrate, there is a great deal of variability in disease expression. There are people with lung disease, cough, trouble breathing, people with GI involvement, people with pulmonary arterial hypertension, and those problems. And how do you say there's going to be one drug that's going to be helpful for everyone? So we are still stuck in the paradigm of looking at primarily one organ system, and then secondarily, if it helps the lungs, well, shouldn't it help the skin as well? And that's not necessarily the case. And there was one other question, 2 minutes.
Is there a place that we can go to register and identify our willingness to participate in studies so that people like you can come through this registry of willing participants and then identify those that may be valid subjects for your study? Because otherwise it seems like it's the other way around.
Yeah.
I have to go to all these different sites and then submit my name, my questionnaire. Is there something where you all go to look for qualified applicants, qualified studies?
Well, unfortunately, currently, no. There is no central registry that all the clinical trials, clinicaltrials.gov will give you a list of all the active trials and a little paragraph that summarizes what they're for and what they're looking for. But still, and usually there will be a contact number with that, but that's trial by trial by trial. It's not something that I could go to and look and try to identify patients. Okay, well, I think that is pretty much the end of it. Well, thank you very much. I really appreciate it.